Hello friends, in this video we are going to learn about the viroids and prions, which are basically related to the family of viruses. And I, John Avinash, pursuing my first MSc Applied Microbiology, will be dealing with this video. In this video, we'll be looking into the contents of various types. First, the introduction, then into the main topic of viroids, its history, definition and examples. Then we'll move on to the prions, its history, definition, examples, and at the end, we will look into the comparison between viroids and prions. In the introduction, we'll see what the virus is. We all know that a virus is a non-cellular particle made up of genetic material, DNA, and a protein that can invade living cells. Beijerinck in the year 1897 coined this Latin name virus, which means poison. The basic characteristics of viruses are they are non-living structures, they are non-cellular nature, they have a protein coat called the capsid, they have a nucleic acid core containing DNA or RNA, they are capable of reproducing only when they are inside a host cell, which means when a virus needs to live, they need a host cell to survive. The first topic, the viroids. Basically, viroids means they are virus-like. That's a key term which we need to understand. History of these viroids. In 1971, Theodor Diner, a pathologist working at the Agriculture Research Service, discovered an acellular particle that he named a viroid, meaning virus-like. Viroids consist only of a short strand of circular RNA capable of self-replication. The first viroid discovered was found to cause potato tuber spindle virus, which causes slower sprouting and various deformities in potato plants. Here is the image which we can see of the PSTV, or also known as potato spindle tuber viroids. We can see the deformities being formed in each potato. Like viruses, potato spindle tuber viroids (PSTV) take control of the host machinery to replicate their RNA genome. Unlike viruses, viroids do not have a protein coat to protect their genetic information. Now into the definition. Viroids are infectious RNA particle without a protein coat. This is the major difference between the virus and a viroid. Viroids are much simpler than viruses. Structurally, viroids are similar or smaller in size, comprising single stranded circular RNA molecules having approximately 250 to 370 nucleotides. Viroids cannot replicate on their own, as their RNA does not encode any gene products. The structure. The viroids are basically made up of two structures. First is the rod-like structure, and the second is a branched structure. Both the structures have a common phenomena, that is single-stranded circle. A few examples we'll be seeing in the few slides. The first one is a potato spindle tuber viroid, which is also called as PSTV, which is typically spread when infected knives are used to cut healthy potatoes, which are then planted. You can see the deformities or the infections that has been caused in the potato. The second is the tomato plant viroid, which is also known as TPMVD, which infects tomato plants, which causes loss of chlorophyll, disfigure and brittle leaves, and very small tomatoes, resulting in loss of productivity in this field crop. You can see the small production, the chlorophyll content is very less, and all the other disfigured which we can witness here. And the third one is the avocado sunblotch viroid, which is also known as ASBVD, which results in the lower yield and poorer quality fruit. You can see here the fruit has been deformed or the infections have been caused in the leaves too. The next example is the peach latent mosaic virus, which is also known as PLMVD, which can cause necrosis of flower buds and branches and wounding of ripened fruits, which leads to fungal and bacterial growth in the fruits. Here you can see the different infections that has been caused due to this viroid. The treatment. Basically, the treatment is the traditional antiviral methods which have been employed for treating the infections caused by these viroids. Second topic is the prions, which is basically a misfolded protein. The history. In 1982, Stanley Prusiner, a medical doctor studying scrapie, a fatal degenerative disease in sheep, discovered that the disease was caused by proteinaceous infectious particle, or he named it as prions. Earlier, the scientists believed that any infectious particle must contain either DNA or RNA, but prions proved it to be wrong since they contain none of this proteinous substance. The, then, because of these proteins, they are acellular and do not contain DNA or RNA. This Prusina's finding were originally met with resistance and skepticism. No one believed actually the initial stages of these prions. However, his research was eventually validated and he received the Nobel Prize in Physiology or Medicine in 1997. The basic definition. Prions are basically the infectious protein or the misfolded proteins. They are normal body proteins that get converted into an alternate configuration by contact with another prions proteins. They have no DNA or RNA. The main protein involved in human and mammalian prion disease is called as PRP, which is also called as prion protein. They are responsible for several neurodegenerative diseases in mammals. These infectious agents are primarily composed of sialoglycoprotein, a combination of sialic acid and glycoprotein, which usually lack nucleic acid. The structure, basically the structure, you can see the structure, the difference here of the normal protein and the disease causing form of prion protein. So the normal protein is named as PRPC and the disease causing form of prion protein is PRPSC. So this is the basic structure and the basic way how they accumulate the PRP. This is a normal PRPC, which is PRPC is normally named as a normal protein, and whereas PRPSC, which is named as the infected or the disease causing prion protein. So they come into contact, they have the interaction, and then they convert into PRP and the accumulation of PRPSC happens. The few examples of the prions are Creutzfeldt-Jakob disease, that is CJD. It is pronounced as Creutzfeldt-Jakob disease. Is a fatal disease that causes degeneration of neural tissue. You can see the normal brain here and the CJD brain here. Second, the transmissible spongiform encephalopathy (TSC) it is included in animals which causes mad cow disease or scrapie in sheep and goats and chronic wasting diseases in elk and deer. The third or the final treatment which we are going to see is currently there is no treatment or cure for neurodegenerative diseases because prions are extremely difficult to destroy as they are resistant to chemicals, heat, radiation, and even standard sterilization process. Now we'll be looking into the comparison between the viroids and prions. First, its definition itself. Viroids are infectious RNA molecules, whereas prions are the infectious protein particles. Second, the terminology. Viroids are virus-like particles. Prions are infectious protein or misfolded protein. Third, size. Viroids are smaller than viruses and prions are smaller even than the viroids. Next, composition. Viroids are composed of single strands of RNA, whereas prions are composed only of protein molecules. Then the infections. Viroids are mostly infecting the plants, whereas prions mainly causes neurodegenerative disease in mammals. The types of diseases that are found in both the types: viroids, potato spindle tuber viroid (PSTV), tomato plant tomato viroid (TPMVD), avocado sunblotch viroid ASBVD, peach latent mosaic viroid ALMVD. And in prions, we see that mad cow diseases in cattle, scrapie in sheep and goats, Creutzfeldt-Jakob disease in humans, Griezmann-Strauser syndrome in human beings. Then the main treatment. As I told you, traditionally antiviral methods have been employed for treating the infectious causes for the viroids, whereas in prions there is no treatment of cure of new generative disease because prions are extremely difficult to destroy as they are resistant to chemical heat, radiation, and even standard sterilization procedures. So the overall table of comparison is here on definition, terminology, size, composition, infection, and the types of disease, treatment, and cure. So thank you for your patient listening and watching this video. For any doubts and queries, use the comment section below. Thank you once again.